Today's presentation is a discussion of leaf fluorescence and how measuring fluorescence enables further exploration of both the light and dark reactions of photosynthesis. Our speaker today is Shannon Loriot, Senior Applications Scientist for LI-COR Biosciences. Joining her today as panelists are Senior Applications Scientists Pat Morgan and Rick Garcia. Shannon's talk is titled, The Importance of Measuring Leaf Fluorescence. Shannon?
Thanks, Ashley. Good afternoon from LI-COR Biosciences here in Lincoln, Nebraska. It's nice to see so many of you joining us today. We are often asked by plant scientists and ecologists who may be interested in or familiar with CO2 exchange measurements, but they often want to know what information can we get from fluorescence measurements. Many plant researchers want to study photosynthesis. They use it as part of their entire suite of other measurements that they use in order to more fully understand a plant's performance under a set of environmental conditions. So this particular webinar was developed for that particular audience. So let's start with the basics. We all know that light energy is absorbed by chlorophyll molecules inside of a leaf. And the molecules are inside of organelles called chloroplasts. And inside the chloroplasts, there are thylakoid membranes that have light harvesting complexes, which are mainly chlorophylls. And once that energy is harvested by the chlorophylls, it is then used in the light reactions of photosynthesis. There are high-energy products, such as ATP and NADPH, that are produced through this process. Those high-energy products are then used in the Calvin cycle to drive the fixation of CO2. So when that light energy is harvested, it is absorbed by a chlorophyll molecule within the sample. And once that chlorophyll has absorbed that light energy, the electronic configuration of the molecule is altered. And this configuration is unstable, and it's typically pretty short-lived, usually less than about 1/100th of a microsecond. And for this chlorophyll molecule to return back to a stable state, it needs to dissipate that energy. And one of the processes it can use to dissipate that energy is either photochemical, shuttling that energy through the electron transport chain, which we just talked about on the previous slide, or it could actually dissipate the energy in a non-photochemical way, such as through heat emission or actually light emission, a red photon of light being emitted by the leaf. Typically, about 95% of the chlorophyll fluorescence that we see under room temperature is emitted by PS2. There is some fluorescence emitted by PS1, but most of the fluorescence that we see is emitted by PS2. All right, to have a little fun and engage all of you participants, we'd like you to answer a polling question. You're going to have a question pop up on the screen, and you'll be able to click what you think the answer is. So here's our first question. When do you think fluorescence was first discovered? A, 1565, B, 1639, C, 1852, or D, 1949? We'll now give you some time to answer the question. If you answered 1565, you are correct. In 1565, Nicolas Menardez, who was a Spanish physician and botanist, first discovered the red glow coming from leaves, and he called this red glow luminescence. Later on, probably a more prominent researcher named Stokes in 1852 is the one who actually coined the term fluorescence. And Stokes is probably more well known for the Stokes shift, which states that absorption bands are shifted to the longer wavelengths when they are emitted. And so he was very revolutionary in this field of work. But in terms of fluorescence, the energy absorbed by a photon raises that chlorophyll molecule to an excited state. And we can see that here in this figure. We can see that in a blue photon of light, there's considerably more energy and therefore the chlorophyll molecule is raised to a higher level, a higher excited state. If the leaf absorbs a red photon of light, we see that the excited level that the chlorophyll is raised to is not quite as high. In between these excited levels, we have relaxation steps. And if the photon of light, the chlorophyll molecule, is relaxed to the first excited singlet state and then relaxes to the ground state, this is the same wavelength as a red photon of light, and this is why fluorescence appears red. In between these other excited states, we can have other relaxation processes going to non-photochemical events like re-emitting the energy as heat, or it could actually be using that energy in photosynthesis in a photochemical event. So you may be asking, well, how do we actually measure fluorescence? There is actually quite a bit of fluorescence coming back from a leaf, and it's bright enough that we can actually measure it some distance away from the leaf surface. But there are really two main types of fluorometers which measure fluorescence. The first type is a continuous fluorometer, and what this does is you need to have a black background or a black room where you have your fluorometer, and there needs to be no other source of red light except the red light that's coming from the leaf. And then this fluorometer has a detector that's specific to the emission wavelengths of fluorescence. And this type of fluorometer is typically used to measure the Kotsky curve, which we see here in this figure. It is basically taking a leaf that's been